Yeah, I want to thank the organizers for having me here. Frankly, I feel like that I'm a spy sent by my comic colleagues, collaborators, but really, it's really great to be here to hear about all this work in Connectome, because my group's interest in topics such as topological data analysis, graph learning, geometric deep learning, and I'm hoping to learn more and see how those can be used to help to solve challenges over there. So this talk will really about a method that started with some topological concept and then combined with computational method, combined with algorithms to lead to a very effective ways to extract hidden graph structure from data. And we first applied, then we first applied this to analyze high-dimensional single-cell RNA-seq data. Okay, so this is most of the work that I'm describing here is joint work with my PhD students, Lucas Magee, and also our collaborator in Alice Institute for Brain Science, Rohan Gala, Uge Sumbul, and Michael H. I just give up pronouncing his last name already. He told me H is good, it sounds cool, so yeah. Okay, and we, needless to say that we're really, in this modern data era, we really come across a tremendous amount of very often very high-dimensional complex data. Okay, and we also heard from the previous talk by Sarah that very often, don't worry, don't be scared about the dimensionality of the data because data often have hidden structures in them and which can help us. In fact, if you think about it, the modern day machine learning, one of the main premises that why it's so effective is because there's hidden structure in data. That's why we can then leverage that data explicitly or implicitly to make predictions, inference, and so on. Okay, so the type of a structure that I am interested in hidden data is basically graph structure, which you can think in mathematical language, you can also think of this as one-dimensional singular manifolds. Namely, each piece is a one manifold, just a simple curve, but they glue together, they have singularities. Okay, indeed, this was originally extended from our work on more general high-dimensional singular manifolds. And so why do I care about it? Originally, the motivation actually coming from that I want to reconstruct the road network automatically from a collection of trajectories collected by your GPS in your car and so on. Okay, and that is a hidden geometric graph. And in the case of neuron imaging, we also know that the neuron cell has the tree morphology and how to reconstruct this tree morphology. This is another graph skeletonization problem. Okay, so what I'm going to do in this talk is that I'm going to first give you a basic algorithm to illustrate the idea how these topological concepts come together with some theoretical understanding to give us an algorithm to do this graph skeletonization in a simpler setting. Okay. Then I'm going to show how this can be extended to handle high-dimensional point clouds data, which essentially we eventually we're going to apply to the single-cell RNA-seq data. Now, I'm also going to bring up one of the concepts because, we're going to find a graph representation of input data, okay? And naturally, we want to compare them and make sense of them, compare such graphs, okay? So I'm going to use one of the concepts to compare graphs called a Weisfeiler-Lehmann distance, okay? So in fact, here, what's interesting about this approach not only is that you can compute it in polynomial time, it's also that this actually treats graph as a Markov model, and you're really looking at compare them through the dynamic behavior of this Markov model, okay? And this also have very interesting connection to graph neural networks, which I will not describe in this talk. Okay, so first, what is this graph reconstruction algorithm I'm talking about? As I mentioned earlier, one of the motivating application is that let's say that I'm giving neural imaging data and I would like to recover either the tree morphology of neuron cells, or maybe I have very noisy whole brain images and I want to summarize the neuron bundles, which summarize them into, say, a graph. Okay? Now, some of the challenges is that this, those data are usually noisy. And also, if you think about this distribution of the signal, let's say that the density indicates the strength of your signal. Okay? This distribution can often be non-homogeneous. Okay, so you could, like if you have tracer injection data, close to the injection site, you have very strong signal, but then far away, you have weak signal, and also sometimes signal just disappeared and then come back, and then you have this little dip in the signal, okay? And if you use traditional approaches, very often rely on local information to make a decision and make inferences, to decide whether I am near a structure or I'm not on a structure. Usually some sort of thresholding is involved to basically tell the signal from the noise. And this makes it very challenging when we have this non-uniform signal distribution. Okay? And in particular, identify the junction nodes very reliably is not a very easy task. So the idea I'm going to describe here, essentially put the two topological ideas together. Number one, I'm going to use, from discrete Morse theory, I'm going to use the global information of your density field, use that to help me to infer the hidden graph. Okay? The advantage of this is that because you're trying to rely on global structure, this helps you to get over those weak signals and also sometimes gaps inside your signal. Okay? The second is that I'm going to bring in persistent homology, which Nick did a good job yesterday to introduce as a way to help me to simplify the obtained hidden graph. Okay? So this originally actually was, this has been a while, the original method was developed actually to, as I mentioned, reconstruct road networks from trajectory data, okay? But this idea of using discrete Morse theory to identify structure in data actually existed earlier, so here's the references. Okay. All right, so conceptually, so here's our first setting, a very simple setting. Let's assume that the input data you give me is already a density field. So I'm given a density field, a function, defined over a cube in the d-dimensional space. I'm drawing the two-dimensional space, but the same algorithm works for any dimensional space. Okay? So now I can view this, I can look at the graph of this function. Okay? So here I'm just drawing this, you lifted it up, the z direction is the function value. Okay? And we want to capture this hidden structure inside. So what's the idea? The idea is very simple. When I look at the graph of this function, I can view this as a terrain. So here's a two-dimensional terrain in high-dimensional space. This is just a high-dimensional terrain. Okay? And we are interested in this skeletons, and you can capture that by capturing the mountain ridges of this terrain. Okay, because the mountain ridges is a place that's locally you're higher in function value compared to when you go off the road, off these ridges. Okay, and that is and the way we're going to capture that, this is where the Morse theory comes in. That we're just going to use a very simple structure from the Morse theory to capture this mountain ridges. Okay, so that's just the conceptual ideas. Now, many of you here know Morse theory much better than I do, and also I'm not going to give you a full introduction here, but just some of the key concepts that we're going to leverage on. Okay, so assume that here I have the graph of a very simple function in R2. Okay, so you have this terrain here. Okay, and given any point on the surface, imagine that the gradient direction of this function, here I'm using a minus in front of the typical one so that I want it to flow downwards. Okay, so I want it to point downwards. So the gradient direction just indicates the steepest descending direction in this terrain. Okay, so now you imagine that you drop waters, your rain is raining on the terrain, they're going to flow down in the steepest, following the gradient direction, okay, because they tend to follow the steepest descending direction, and then it's going to trace out a curve, this flow line called the integral line. Okay, and you can also imagine the raindrop climb uphill, and this extends to a curve. Okay, when would such a curve start or terminate? Well, it has to start and terminate in places where you don't have any gradients. The gradient vanishes. Okay, those are the critical points of this function. Okay, so now in general, if most of this integral curve terminates in the minimum, Okay? But sometimes in this two-dimensional case, I only have one type of saddle points, this little green dots here. Sometimes they terminate at saddles. And this is necessary because if you imagine that this is basically, if I have a ring job here on this side, I'm going to go to this basin, this minimum. And if I'm going to the other side, I'm going to the other basin. Somewhere in the middle, you have a transition. And that's when you have this integral line ends, intuitively ends at a saddle point. Okay. So this is a type of integral line I care about. I'm going to look at the integral line going from maximum to the saddle and between the maximum and saddle. Okay? This gives me a little network which forms these mountain ridges. And in high dimensions, this is so-called one unstable manifold that connects the d index d critical points, the maxima, with index d minus 1 saddle points. Okay? So that's essentially is the idea. And now conceptually, we see that this mountain, one unstable manifold indeed captured the mountain ridges because when I go to two sides, this curve is separating basins from basins. That's the middle line separating the basins. Okay. All right, so this is just one from one of this neural imaging data. Now, this is great. And this is the conceptually in the smooth case, okay? When we have a nice function, differentiable function, and we can define this way. But of course, we know that in practice, we don't have that. In practice, we have discrete data. Our domains are usually either triangulated, like what Nikki was describing yesterday, with some components, triangles, tetrahedrons, and simplicial complex, okay? Or they can just be a cubical complex, okay? So we have this discrete setting. And now, this one unstable manifold is related to the gradient. So it's a differential object. Okay? So you don't want to really numerically estimate that in practice because it's not very stable. Simplification is the issue. In fact, you can do that in 2 dimensions, but when you go dimension higher than 2 dimensions, simplification becomes very challenging as well. Okay? So what do we do here? Well, Robin Foreman had this really beautiful work on discrete Morse theory, and this is not a discretization of Morse theory in the piecewise linear setting. Okay, this is actually a combinatorial analog of the Morse theory, and he proved many beautiful results, saying that in this setting you can still have this criticality concepts and which behave very similarly to the standard in the smooth setting. What critical points is doing in terms of the homology group of the sublevel set and so on. Okay, so why do we like this is that number one, I'm now having a combinatorial objects now. Okay, I'm going to, now everything is numerically more stable. And also number two, there's also a way to simplify this combinatorial in this combinatorial discrete Morse theory, which you will see later that it works much better, it's very robust in practice. Okay, it works in any dimensions. Now, as again, I don't have time to really describe the discrete Morse theory in detail, just a high-level intuition. As I said, now your input is actually, let's say for simplicity, let's say we have a simplicial complex as input, but it doesn't have to be simplicial. His original work is also on more general cell complexes. Okay, so now, given this simplicial complex K here, a discrete gradient vector is not a real vector. It's actually a pair of simplices. It's a pair of simplices, say a triangle and an edge that's an edge and a triangle incident on that, or a vertex and an edge incident on that. Okay, so in general, it's a P simplex together with a P plus 1 simplex incident on this P simplex. Okay, so even though this is not really real vectors, but the way I plot this, I hope you can see, you can still have this concept of flow direction, right? Because, for example, I can start from this edge and flow into this triangle, and I can choose this edge to go out and flow into this triangle and continue. So you still have this concept of flow paths, okay? And a discrete gradient vector field is just a collection of such pairs, as long as no simplex appear in more than one such pair, because at any moment, you should have only one gradient, okay? The second is that if I follow this path, I just said this kind of flow path, I should not have cycles, because gradient should always just flow down or flow up, whichever way you like it, Okay, so if you have such a collection of pairs, this discrete gradient vectors that satisfy these two conditions, you get a discrete analog of the gradient vector field. Okay, and critical simplex corresponding to critical points now are just those simplices who didn't appear in any pairs, namely gradient vanishes there. Okay, so the pink colors I made here are the critical simplices. Now, this is really very high level, but I'm very hand-waving. Let me just tell, yes?
Is there some rule to assign the vector field to the simplest?
The only condition for the discrete gradient vector field is the two conditions I give here. Anything would be a valid one. You can have good ones and bad ones. Same as if I give you a space, R2, you can define different function, which would induce different gradient vector field. Okay, yeah. All right. So very hand-wavingly, if you're in d-dimensional space, let's say that you now have a triangulation of a d-dimensional hypercube, okay, you can roughly think that critical k simplices corresponds to index k critical points, okay? And critical, for example, in this example, my domains are R2, critical edges essentially is index 1 critical points, which is corresponding to saddles. And the critical triangles, that's index 2, which is the maximum, and the critical vertices at the minimum. Okay. And earlier we said that we care about one unstable manifold going from max to saddle. So in this case, this corresponds to a flow path going from triangle to critical edges. Okay. But note that if my domain is not R2, it's RD, then this one unstable manifold would become a path going from d simplices to d minus 1 simplices alternating, which is more expensive to compute and to maintain. So I'm just going to do a small trick. Let's imagine I look at the function minus f. So instead of one unstable manifold, now I look at one stable manifold, which is going in between. No matter what the dimension of your domain is, this is always going to be between vertices and edges. So you're going to look for paths alternating between vertices and edges. Does it make sense? So now you're looking for paths going from minimum to saddle and to minimum, okay, because you invert your function. And one of the useful operations is this Morse cancellation, which allows us to reduce the complexity of your gradient vector fields, to reduce the number of critical points. In particular, you can cancel out a pair of critical simplices if there's a unique path connecting them. And the way you cancel it out, you just locally invert the orientation of all this pairing. So it's quite easy, okay? So it's purely combinatorial. And you need to, this is kind of analogous to this Morse cancellation in the smooth domain where you cancel out a pair of critical points whose criticality differ by, index differ by 1, okay? And you do need this condition of that there's a unique path, because otherwise, if you invert the orientation of 1, you're going to form loops, just so that you always maintain you have a valid discrete gradient vector field. Okay, so that's it. So that's how you can smooth out, simplify your discrete gradient vector field. So, all right, great. So we now said that, okay, I'm going to look at this combinatorially, and I have this discrete gradient vector field, and I also know how to simplify them to make it simpler. But we didn't really say that I have many, so the cancellation can cancel out any pair of critical simplices as long as the condition is satisfied, there's only a unique path between them. It doesn't tell me which pair I should really cancel out, okay? Further, note that so far, the description of this discrete Morse setting, I didn't even talk about the original function that we may have in mind. That has to play a role here, right? So the intuition is that I should cancel out only those pair of critical simplices. That's not important with respect to my function, okay? And to this end, this is where the persistent homology comes into picture, which will kind of guide me what pair of critical simplices to cancel. Okay. Now, we already heard persistent homology introduction from Nikki yesterday using the simplicial complex setting, and earlier in Smita's talk, she also mentioned this. So here, let me actually look at this from a function setting. So persistent homology essentially just that instead of looking at homology of a fixed space, I imagine that I have a way to grow my space. That's called a filtration. Okay? And as the space gets bigger and bigger, I track what happens to the homology groups. Okay, and the percent homology gives you a specific way to relate the birth and the death of the new homology classes. Okay, so in this function setting here, I have a really trivial example, just a one-dimensional function. Okay, so this is a graph of the function. And the way I'm going to grow this domain is that I'm simply going to sweep the domain bottom up in increasing function value. And at any moment, I look at the so-called sublevel set. This is the red portion that I draw here. Okay, then I want to track the homology, the change of the homology here. Since our function, the domain is so simple, it's just R1, here I can only talk about the zero-dimensional homology, and beta 0 is the number of connect components. So you can think that I'm going to grow this function and then keep track of connect components. Okay, so when does this change? This change when you sweep past a critical point. Okay, in particular, when you go past the minima, you create a connect components. And when you go past the maximum, you merge a connect components. You can think the minimum creates a zero homology, and the maximum kills it in this 1D case. Okay, and when some connect components is killed, essentially percent homology dictates there's a unique way, canonical way to decide what is the feature killed by this critical introduction of the critical points. In particular, in this case, you get that X3 is paired with X4, namely X4 kills the little components created the X3. Okay, and when you finish the process, you get a bunch of pairings, and now note that these pairings are between critical points of your function. Okay, and each of the pair indicates the birth and the death of some, in this case, zero-dimensional homology, and the high difference, the so-called persistence, indicates the importance of that. Okay, so this is just in the smooth setting, and again, we have piecewise linear setting, but no worry, you can use lower star filtration to approximate this sublevel set filtration that I was talking about, okay? And in that case, there's an algorithm called the persistent algorithm which essentially computed this pairing. Now the pairing is between simplices now, okay? So in particular, between a simplex of dimension p with a simplex of dimension p plus 1. And just in case that you are interested in topological data analysis, my collaborator Tamao Day and I, we have a textbook on computational topology published 2 years ago. And you can download a PDF version on my webpage. Okay, so now we have all the ingredients, and we're ready to have our first algorithm. And it's quite simple. Suppose we're given a triangulation of our D and some function given at vertices of this triangulation, okay? I just simply first compute the persistent homology, use the persistent algorithm to compute the pairing of all simplices. And at the beginning, I have a trivial discrete gradient vector field. There's no pairs at all. Then I use, given the threshold, percent threshold delta, I basically cancel out all the persistent pairs of simplices whose persistence is smaller than my threshold. Okay, that's it. Afterwards, I pick the remaining critical edges. Remember, critical edges corresponds to saddle points. Okay, so I pick the remaining critical edges whose persistence is bigger than delta, and I trace out the one stable manifolds in my domain. So, and you actually only need two skeletons of the domain in order to carry out everything, no matter what's the dimension of your domain, because your final one stable manifold is between, you only need the vertices and the edges. But to do the computational persistence, you also need a triangle. So you only need two skeletons of the domain. Okay, you don't need a higher order simplices. And of course, you can convert this one to handle cubic complex, which is more common in imaging data. So that's it. That's the entire algorithm. And this is just some, really simple example. As I told, this algorithm originally was very old. We developed more than 10 years ago. And this is from the DIDAM data challenge. So this is not the challenging data, and the many other methods works very well, but I'm just showing a proof of concept that this is a skeleton that computed by the persistence. This is another data set. There are actually two neurons, and which will capture both. And this is when we apply this to the whole brain imaging from my collaborator Pasamitra from Cold Spring Harbor lab to summarize some very coarse tracer injection data. And this, just to give you another application, this is on road network reconstruction from trajectories. And you see all this. This is trajectories from Berlin. This is automatically reconstruct the road network. And so this is, it's not visible here, but this is highly non-uniform close to your downtown area. The density is so much higher than the other places, so you don't want to use just the standard density threshold to clean up your data, okay? And so I can see that this captures the structure, multi-scale structure pretty well. I'm using this one just to give you an example, to show the intuition, the power of the topological methods coming from. So this is, before us, the state of art for road network reconstruction. What happened, you see that you see broken pieces. Okay, and the power of a topological method is because now you're using mountain ridge. This is a global structure. You may have a little gaps, but I'm still going through that to connect to other max significant features. So that's why you get a much better global connectivity. Okay, so that's one of the main intuition. All right, so now, in fact, after we had this algorithm, it's very intuitive. It came intuitively to us, but then why should it work? What's the theoretical justification? Okay, in fact, we're putting two things together, discrete Morse theory and the persistent homology, and it's not clear that they really match to each other. Okay, so we try to prove that some guarantee that the graph we reconstruct should be close to the true hidden graph. Okay. And then when we try to do that, we did get some theoretical results, but when trying to do that, we actually also realized the connection between the discrete Morse theory and the persistent homologic algorithm, which further simplify this algorithm tremendously, which is already conceptually very simple. So in particular, here, the second step right now, I need to take all those pairs given to me by persistence, saying they're not important. And then you check whether they are indeed cancelable or not. And if they can be canceled, I should cancel them, doing this, modify my discrete gradient vector field. That's what we're supposed to do, okay? And it turned out that you don't need to do that. It turned out that after the persistent algorithm, all you need to do is that you just look at the so-called negative edges, which essentially is those kind of edges that destroys homological features, okay? You collect all of them whose persistent at most delta. This is going to give you a spanning forest of your input points. And the spanning forest, so this scan, this is just a linear scan. But this spanning forest contains, you are not maintaining any discrete gradient vector field anymore. All you have now is the spanning forest. And this spanning forest is sufficient for you to retrieve the one stable manifolds in the third step. Okay? So the algorithm, you just need to do some tree traversal. So essentially, after the so this is a new algorithm. The first step is persistent computation, which is a black box that we have to use, okay? And then once you finish with the persistence, you just collect all this linear scan, all these negative edges, as I said, and then pick all the remaining critical edges persistent bigger than delta, and do certain tree traversal to get your one stable manifold. That's it, okay? And the algorithm takes linear time once your persistence is computed. Now, persistence is expensive. We also heard yesterday. The best algorithm is matrix multiplication time, but in our case, we only need to up to 2 skeleton. Okay, and there's also a lot of work that's been devoted from computational topology, try to speed up the persistent computation in different settings. Okay? And this algorithm will also show that under some certain noise model, you can actually show that the reconstructed graph is both geometrically and topologically close to the hidden graph that supposedly data is sampled from. Okay? And this holds for any dimensions and quite easy to implement now. You just need to compute the persistence. Let me just mention that the way with Partha Mitra's lab, we did also try to use this to do the cell processes detection in 2D images. The idea is the following. The idea is that the standard segmentation, state-of-the-art machine learning segmentation tools, such as UNet, which is based on CNN, is that they capture signals very well, but they can miss, when you have broken places, they can miss the connectivity because it's not in their loss. They don't care about the structure, okay? And now, using, oops, I think it's the wrong order. Using the discrete Morse theory, we can capture this global structure pretty well. But the problem is that if your input image is noisy, we got a lot of noise as well. Okay, so the idea is that how about we put them together? We use one to augment the other. So that's this machine learning pipeline that we have, that you basically try to use both so that you can use CNN, try to remove the noise from DM, and use discrete Morse graph, try to give you a more global structure.
But isn't the discrete Morse graph actually an unsupervised method, whereas the
it is unsupervised? Right? Yeah. So we actually put a discrete. So in this approach, we put a discrete Morse graph as another channel for input. Okay, yeah. So, but in a different collaboration with Chao Chen from Stony Brook, we use that as in the loss to guide it. So that's a different approach. In this work, we put as input. Okay, I just want to mention this work. All right. So that's the base algorithm. But as I said earlier, we ultimately want to use this for RNA single-cell data. And so here, you can think that your input is a bunch of points in high-dimensional space in RD, and you try to construct a hidden graph skeleton from this points data, okay? And you might say that, okay, what's the problem? Sounds like this is, well, I mean, the first thing is that now we have points, okay? Points, previously, our algorithm required that I have a triangulation of the domain of interest, which is RD, a cube in RD. So now we just have points. How do I get some description of the domain of interest? Okay. Now this we heard, there are many choices, and Nikki mentioned the RIPS complex yesterday, which essentially is that you say, okay, you can just take all the points that are close enough to each other and connect them, and then take the induced, and if 3 points are connected to each other, you put the triangle in, 4 points are connected to each other, you put the tetrahedron in. To get this essentially is almost like you're growing a ball of certain radius around each of the point and look at the space of this union of balls. Okay, so not the RIPS complex, a related concept, the check complex will give you the nerve of this union of balls, and the RIPS is related to the check. Okay, so this is a very popular thing that people do in practice to get the space. And once now I have this triangulation, the simplicial complex of the space, I can just run the algorithm I told you earlier. I can estimate the certain density at each of input point, and use that as a filtration, and then construct a hidden graph. Okay, so I have a domain, I have a function, and then I can get an algorithm to reconstruct the graph skeleton. Okay, so let me call this baseline algorithm. Okay. Now, what's the problem of this approach? Well, one of the biggest problem is that the data distribution can be very non-uniform as well. Okay, certain clusters, the points are very close to each other. Some other times they can be very far away from each other. Okay, so what is the right scale to use to build this RIPS complex? Okay, so here I'm showing a few examples that if you use different R value, this is a reconstructed graph, and if it's too low, I don't yet connect it. If it's too high, I may connect it, but I also may have wrong topology there. So this is a very simple one. In fact, here's another one that I'm just, this is an artificial example just to make the point. You have a small loop here, which is very dense, and you also have another circle there. Okay, you see that there's no good scale, because if I take the R small, I can capture the small one pretty well, my simplicial complex. But if I use R to be large, well, after this one is connected, the small little circle now got filled, completely filled by the RIPS complex. So you don't see that the loop inside anymore. Okay, so there's no good scale for this case, and the bottom one is the reconstruction using the baseline algorithm. So it turned out that we're thinking about, we want somehow our algorithm to directly handle not just a fixed scale simplicial complex, but a filtration, okay? And there's a simple way to modify, which I'm going to show you the modification in a minute. Now, the nice thing about this modification is that, number one, actually, we can indeed provide some theoretical understanding of what is the output now being computed by this new DM algorithm. And also now, we can use any filtration we like. We're going to use one, which is called specified weighted RIPS, which essentially, for very high-dimensional data, we heard that RIPS can be very expensive as your radius become large. This one brings in specification. As your radius become larger, you can also look at your data at a coarser scale. So it brings in this specification. So you so-called the specified weighted RIPS filtration. So we can now use this filtration to this new algorithm. Okay, so it's a handle, the size is not too big, but now I can also look at all scales now. Okay, and the theoretical guarantee of this approach also indicates that we're still getting meaningful feature in our reconstructed graph. Okay. All right, so what's the modification? Previously, I have a domain and a function, okay? And then we look at the persistence induced by this function. Okay. Now, note that once the persistence is computed, I got a bunch of pairs between simplices. The remaining two steps doesn't even care what's your persistence, what you use. I just care that you give me a bunch of pairs of simplices, what's importance, that's a persistence value. Okay, then I have a spanning tree, I just do a linear scan, get a spanning tree, and I do some tree traversal to get to the one stable manifold. Okay, so we don't have to use that domain, we can use any so-called simplex-wise filtration, namely, it's a sequence, an ordering of simplices, that's it, okay? And I can compute the persistence algorithm induced by this ordering. You basically add one simplex at a time, this ordering, okay? The remaining two steps maintains the same. What you can say is that, let me not introduce this definition, what you can say is that the output graph, if you look at the cycles inside, so here this result doesn't quantify the tree part of your graph, only the cycles, okay? So the cycles in your graph actually corresponds to the so-called lexicographic optimal persistence cycle basis for all the points in your persistence diagram. That's important. In this example, this is red line is the threshold. I only have one important persistence point. In general, there could be multiple one, okay? So this graph is going to contain generating cycle for those important persistent features. And the generating cycle for persistent, for homology class is not unique, okay? But this is going to pick the so-called lexicographically optimal one, namely the one that in my ordering appears as early as possible, okay? Now, if your ordering, the input ordering of simplest is such that important ones comes first, what I was saying here is that I'm looking for a representative cycle that is using edges that's as important as possible first. Okay, so that's basically what this is saying. Okay. I'm just wanting to show you that in this example we saw earlier, this is the output. And here, this is the very, for those of you in TGDA, we know this is a very classical data set by Gala on 50,000 3x3 image patches, which supposedly should have 5 independent loops. And this is our method, and it actually gets the structure exactly as originally described. And this is using the baseline. Okay? All right. Now, okay, I think I should really hurry up, but let me just say that we have, all right, now, what I told you just now is that I give you a way, you give me a point set, now I kind of represented, use a graph to represent the data point, okay, the point clouds data. So with this graph representation, what if I want to compare them? If I give you 2 data sets, I want to compare them. How do I compare them? So now I need to be able to compare graphs, okay? And I think this was also mentioned earlier yesterday in some of the talks. Graph comparison is actually highly non-trivial, okay? The typical distance for graphs, like graph added distance, these are all NP hard to compute and also to approximate, okay? In fact, even when you go to tree added distance, that's NP hard to approximate, okay? So, now, at the same time, comparing graphs related to the graph isomorphism testing, okay, namely give you 2 graphs, you want to tell me whether they're the same or not. Okay. So here's a simple procedure which essentially takes linear iterations to tell, it doesn't solve the graph isomorphism problem, which we know that take a super pseudo-linear polynomial time to solve, okay? And, but it can differentiate most pair of graphs. Okay, namely, the pairs that it cannot differentiate is only of measure 0. Okay. So this, so-called WL graph isomorphism test, if you give me 2 graphs, in linear iterations, I tell you either they're not the same, or I tell you that I cannot tell it's potentially isomorphic. Okay? And how does it work? So it's a very simple idea. let's say that now we're given two graphs, and you view them as attributed graph. Namely, every node also has some attributes. If you don't have anything, just use constants. Doesn't matter. Okay? Here, I'm using degree as the initial attributes of the nodes. Okay. You give me these two graphs, you want to compare them. I say that, let me look at the collection of features, the degree distribution. If they're the same, I say if they're not the same, the two graphs are not the same. But if they are the same, then I cannot tell, right? So if they're the same, like in this case, the degree distribution is the same. So then what I do is that I say for every node, I'm going to now aggregate all the features of my neighbors and use this entire collection as my new representation of who is my feature. So for example, for this node, now my representation is 1, 1, 2, 3. So I use the multi-set of the features. Okay? This one now is 2, 2, 1, which is now, they are different now. So now you can tell them these two graphs apart. If you can tell them apart, you return. If you cannot, you continue. After linear iteration, guarantee that this procedure will stabilize, will not change the color set anymore, then your algorithm terminates. Okay? Very simple algorithm. And you can also think that what I'm really doing is that for every graph, I build this hierarchy that at every step, I keep updating each node have a different feature representation. Okay, the feature representation keep getting more and more complicated. And in particular, as I aggregate the features of my neighbors, my feature representation becomes a multi-set. And later, as I aggregate more, it becomes multi-sets of multi-sets. Okay? And then when I compare two graphs, I just compare this two collection of feature sets across all scales. If any scale is different, they are not the same. If they're all the same, I cannot tell. They're potentially isomorphic. Okay? All right. And this is only telling whether the two graphs are the same or not. But we want to compare them. We want to have a distance measure. Okay? So the idea is that you can basically kind of metrize this multi-set. The reason right now it's only yes or no is because you represent features at any moment using a multi-set of features of the neighbors. Okay, I want to convert this multi-set to be measures. Okay, and this aggregation turned out that I can use the push forward in suitable spaces to model this aggregation, and this hierarchical probability multi-sets of multi-sets now become hierarchy of probability measures where the space gets more and more complicated, and at any moment I can compare them using optimal transport. Okay, so I'm skipping a lot of details under the rug, but you have certain theoretical guarantee of this distance measure as well, and you can compute it in polynomial time. And this is also connected to the popular method passing graph neural network and can be used to model the universal approximation of that. Okay? And I want to point out that this updating of the feature is really viewing the graph as a Markov model. I'm using that Markov kernel to do the aggregation. So this also can be used to compare Markov chains. All right, so I think in the next 5 minutes, just give me 2 extra minutes. Let me just tell you that how this is finally applied to the single-cell RNA sequencing data. So one of the things I want to make is that very often, given this high-dimensional data, when we try to make sense, we want to bring down the dimensionality. And often after we bring down the dimensionality, we focus on cluster structure in data. Okay, and the point I'm using graph, what we're trying to make is that you also want to look at the continuous space in your data, which of course in the previous talk by Sarah, also there was this continuous trajectories. Okay, so what we're going to do is that I want to argue that with this high-dimensional RNA-seq data, okay, without bringing down the dimension, just in the original raw space, I want to use this graph skeleton to represent them. And I want to argue this is meaningful, this indeed capture information, okay? And then once we argue that this is a meaningful representation, I want to use that to provide analysis of those single-cell data. So the data set we have is from Allen Institute. This, basically, we have about 1 million cells labeled in 18 different regions in our brain, okay? So for each of the region, I'm going to take about 10,000 cells just randomly, and I'm going to take 10 such random subsets. You can think of that as a sampling of my big data sets, Okay, so altogether, this would give me 180 total subsets. And for each such 100,000 cells, I would do this DM reconstruction, get a graph representation. So this is just to show that this graph representation actually indeed captures the key information that appears. So what we're showing here is that if you now look at the data in that collection and look at, if I look at the so-called KNN coverage, namely, I look at the KNN of any cell. If that's one of the k-nearest neighbor is in my graph skeleton, I consider that my graph skeleton captured that data. Okay? So this red dot is this KN coverage using the discrete Morse graph, which I didn't put a number. It's only between 200 to 500 cells. Okay? It captures most of the points. Okay? But if you take a random subset, so we took many random subsets and look at the KN coverage, that's pretty, this coverage rate is very bad. Okay? That means that even though I have a significant reduction in the number of cells, but now it seems that the cells, the skeleton still cover well this entire collection of cells. Okay? This is just to show it's quite stable with respect to K as well. And the second interesting thing is that we want to show that using this graph representation, it also indeed is a minimal way that it can actually differentiate between different brain regions. Okay? So here, remember that I have 18 brain regions. Each region, I have 10 random subsets of cells. Okay? Now, between this, the 180 collection of point clouds, I can compare them in a different way. I can compute the discrete Morse representation and then compare them using the WL distance I just talked about. Or I can just directly take some subset and do the Waterston distance, treating them as distribution and compare the point set directly. Or I can look at the Waterston distance, optimal transport distance, between all the cells in my discrete Morse skeleton. Okay? And to know that, so each of this distance measure would give me a distance matrix, which is 180 by 180. Okay? Now, this 180 data set is coming from 18 regions. What you want to see is that you want to see that those data sets coming from the same region, you want them to be closer to each other than others. Okay? So for this, we have the so-called triplet consistency score. This is inspired by work of Sanjoy Dasgupta in modeling the hierarchical clustering. Basically, I said that for any 3 data sets, if A, B are coming from the same region, C is from a different region, then my distance between A, B should be smaller than distance between A to C and B to C. Okay? And this is called the triple consistency. And I want to look at all such triplets and see that what is this rate, frequency of this consistency. Okay? Now, so here just the results, just putting the punchline is that this worst doesn't mean that this behaves worse. This worst means that when we do different random sample, I pick the worst score for the Morse, but they still outperform any of the other methods. As you can see, the triple consistency is very close to 1. 1 is the best you can get. And I think that I'm really out of time. Let me just also tell you that now I have 2 minutes. You can use this structure because now you have this graph representation. So how can we leverage this representation? So what graph gives you that the clustering is not giving you is the graph gives you the continuation of how your space is changing. Okay, even between 2 different clusters, it gives you a pathway connecting them. Okay, so here we're looking at that. If you look at the branches, and this is labeled by the cluster types of cells, we want to see that how stable are the type of the cells. Okay, so to measure that stability, we use this 1 LCL. You can just think that the higher this number is, that means that my neighbors are all same type as me, basically. Okay, and so what I'm saying here is that, so first the point is that I'm using a very low percentage threshold, so you see the graph very simple, a simple graph, but the graph can separate the different classes of the neurons very well, and it also separates the subclasses pretty well. Okay, and if you look at how does this consistency change, what's interesting is that it seems at the beginning, which you basically have a lot of information because you didn't use the persistence threshold, so you have the entire data sets. This consistency is not good, namely it's very noisy. But once you increase the persistence, you get a pretty high value, like kind of a denoising. You have a meaningful skeleton where this means that the subclasses, most of them are well separated because you don't keep jumping between different types. Okay, and so let me actually just move on to here. You can also use this to look at differentiation between different types of the cells, but in different regions. Okay, interestingly, like so here I'm showing this SST 99, which is one of the cell cluster type, and in one of the region, it's pretty consistent, like they are clustered well together. But in a different region, they're pretty bad. They keep, like that region here, it keep mixed with different other types. It's hard to differentiate between this type and other type, okay? And so you can use this graph to help to do that analysis as well. And let me skip this last one. And just come to my concluding remarks that I hope that this, even though this is not about connectome data, but I hope that some of the methods that are used here can be useful for that as well. And just a very high-level perspective, which is that our humankind have always been using mathematics as a tool to help to understand the world around us, okay, doing computations, often by hand, historically. And I think in the modern age, when we have this unprecedented amount of data and also this amazing computation power, the geometric topological data analysis, which is the field that I'm working in, kind of bring the new perspective. How can we combine this classical mathematical concepts and ideas with algorithm development, with the consideration of data to make them more effective to handle the complex data? And neuroscience is provide a very rich platform for this kind of ideas because neuroscience is very rich in structure, implicit or explicit, static or dynamic. So it's really a very nice platform for geometric and topological data analysis. Thanks for your attention.